What is recombinant DNA and how is it made? All that and more will be answered later in the video. But right now, let me just brief you about DNA and its structure. Deoxyribonucleic acid, or in other words, DNA, is made up of nucleotides, which are subunits of nucleic acid basically. Those nucleotides are shaped like a double helix. The DNA structure was discovered by the molecular scientists James Watson and Francis Crick, but every person has their shadow. This here is the molecular scientist and biologist Rosalind Franklin, or as you may know her as the Dark Lady of DNA. As to why Rosalind Franklin got that nickname was because she was the first to take a photo of the DNA as a double helix. She took the famous photograph, Photo 51, of DNA. It demonstrated DNA as a double helix for the first time. And as you can see here, this is Photo 51. Moving on to the structure of a DNA nucleotide. It would be brief though. A DNA nucleotide is made up of a 5-carbon sugar, a phosphate group, and a nitrogenous base. Now, without further ado, let's get into recombinant DNA. But before we get to the scientific details, let's just brush up on a little history. The history of recombinant DNA dates all the way back to the early 1970s, specifically speaking 1972, because the Stanford biochemist Paul Berg performed the first production of rDNA during that year, and henceforth the first application of rDNA on a patient was in 1974 by Stanley Cohen of Stanford and Herbert Boyer of UCSF. Have you come across the term genetic engineering, or perhaps biotechnology? In simple terms, gene manipulation. That's the meaning of rDNA technology. rDNA, as you might have already guessed from its name, recombinant, is DNA that has been tampered with. It's artificially made by combining the DNA of different organs. Moving along to how rDNA is made. There are 6 steps in total involved in the making of rDNA. First is isolating the genetic material. Second is the cutting of DNA or a restriction enzyme digestion. Third is using a PCR for amplification. Fourth, the ligation of DNA molecules. Fifth, inserting the recombinant DNA into a host. And last but not least, the isolation of recombinants. As I've said before, the isolation of the genetic material or DNA's isolation is the first step. DNA is isolated in its pure form, which means they are devoid of other macromolecules. In rDNA technology, the initial step to extract the desired DNA in its purest form, that is, free of extraneous molecules. Because DNA coexists with other macromolecules such as RNA, polysaccharides, proteins, and lipids within the cell membrane, it must be separated and purified using enzymes such as lysozymes and cellulase. Other enzymes or treatments can remove other macromolecules. The DNA eventually precipitates out as fine threads as a result of the presence of ethanol. After that, the pure DNA is spooled out. As for the second step, which is the cutting of DNA, the restriction enzymes are quite vital. It helps to identify the location where a designated gene is introduced into a vector genome. The said reaction is known as restriction enzyme digestion. They entail incubating pure DNA with a restriction enzyme of choice at conditions that are appropriate for that enzyme. The agarose-gel electrophoresis technology displays the restriction enzyme digestion's progress. This method entails passing the DNA across an agarose gel. When current is applied, negatively charged DNA flows into the positive electrode and is divided into different sizes. This permits the digested DNA fragments to be separated and sniped out. The same method is used to process the vector DNA. If you don't know what is agarose gel electrophoresis, just as I didn't before, it's a method of gel electrophoresis used in biochemistry. And gel electrophoresis is a method for separation and analysis of macromolecules and their fragments based on their size and charge. Now, as for the third step, an amplification of DNA. Copies of genes are made through PCR or polymerase chain reaction. It is essentially a process to increase a single DNA copy into several copies after the desired gene of interest is cut with restriction enzymes as per the second step. It allows a single copy or a few copies of DNA to be amplified into thousands or millions of copies. The following components are used in PCR reactions that are conducted on thermal cyclers. If you don't know what thermal cyclers are, they are laboratory apparatus used to amplify segments of DNA via the polymerase reactions. They are also referred to as thermal cyclers. The first component is template, in which DNA has to be amplified. Second are the primers, in which the oligonucleotides are tiny and chemically produced. And oligonucleotides that are complementary to DNA region. If you don't know what oligonucleotides are, they are a polynucleotide whose molecules contain a relatively small number of nucleotides. And third component is the enzyme, and basically it's the DNA polymerase. And last but not least are the nucleotides themselves. The enzyme is required to lengthen the primers. PCR can be used to amplify the cut DNA fragments, which can subsequently be ligated with the cut vector. And as for the third step, or amplification of DNA, copies of genes are amplified through PCR or polymerase chain reaction. It is essentially a process to increase a single DNA copy into several copies after the desired gene of interest is cut with restriction enzymes as per the second step. It allows a single copy or a few copies of DNA to be amplified into thousands or even millions of copies. The following components are used in PCR reactions that are conducted joining DNA. The vector and the section of DNA are joined in this step. It is achieved with the help of the enzyme DNA ligase. With the same restriction enzyme, the pure DNA and the vector of interest are cut. This yields the cut DNA fragment and the cut vector, both of which are now open. Ligation is the process of putting these two parts together with the enzyme DNA ligase. The resulting DNA molecule is a hybrid of the interest molecule and the vector DNA molecules. Recombination is the term used in genetics to describe the merging of different DNA strands. As a result, this new hybrid DNA molecule is known as a recombinant DNA molecule, and the process is known as recombinant DNA technology. Step 5. Insertion of rDNA into a host. Here, rDNA is added to the recipient host cell, and the entire process is called transformation. Post-insertion, the recombinant DNA multiplies and manifests as manufactured protein under favorable conditions. The recombinant DNA is then transferred into a recipient host cell, most commonly a bacterial cell. In this stage, the term for this procedure is transformation. Bacterial cells have a hard time accepting foreign DNA. As a result, they are given treatments to make them quote-unquote capable of accepting new DNA. Thermal shock, Ca++ ion therapy, electroporation, and other procedures may be applied. And now finally, the last step, recombinant cell isolation. A mixed population of converted and non-transformed host cell results the process. Only the transformed host cells are filtered during the selection procedure. The marker gene of the plasmid vector is used to distinguish recombinant cells from non-recombinant cells. And finally, these are some uses and aims of recombinant DNA. rDNA technology has been widely used in medical science industries and agriculture. The following highlight the application of rDNA technology in brief: producing recombinant HB vaccines, producing human insulin, facilitating better crop production, producing growth hormones in humans for treating dwarfism, better gene therapy, acquiring DNA fingerprinting, and diagnosing several types of disease.